Hi, my name is Brian Kim, Professor and Vice Chair of Research in Dermatology, Director of the Mark Lovell Center for Neuroinflammation and Sensation at Icahn School of Medicine at Mount Sinai. In Vignette 4, I'll be talking about itch signaling and neuroimmune crosstalk. Itch underlies atopic dermatitis, prurigo nodularis, and chronic spontaneous urticaria, and is one of the most bothersome symptoms for patients with a substantial negative impact on their quality of life. In all three of these chronic pruritic inflammatory conditions, itch involves neuroimmune dysregulation that may lead to neuronal sensitization and itch pathway activation. However, itch quality varies across these conditions. To understand this, one must appreciate the molecular and cellular basis of specific neuroimmune interactions. Itch sensation occurs when pruritogens, such as neurotransmitters, for example, histamine, bind to receptors on itch sensory nerves in the skin. Historically, itch sensation has been divided into two major pathways, the histaminergic and non-histaminergic itch pathways. However, this distinction is largely imbalanced. Non-histaminergic itch is responsible for the majority of chronic itch types and can be mediated by a variety of molecules, including allergens, cytokines, leukotrienes, proteases, and extracellular matrix proteins. Let's look first at the transmission of itch signaling using a well-recognized pruritogen, histamine, as an example. The histaminergic pathway is initiated when IgE activates the cell surface high-affinity receptor on mast cells, Fc-epsilon receptor, in addition to basophils to release histamine and other itch mediators such as leukotrienes, which contribute to acute itch. Once released, histamine binds to H1 and/or H4 receptors on histaminergic nerves to trigger an electrical impulse within itch neurons or proreceptors towards the spinal cord. Itch is primarily mediated by unmyelinated C-type nerve fibers in the dorsal root ganglia, which transmit signals to the spinal cord via neuropeptides such as brain natriuretic peptide, or BNP. Here, the activated spinal neurons can release other neuropeptides such as neuromedin B or gastrin-releasing peptide, which bind to their respective receptors within other neurons in the spinal cord. After passing through the spinal cord, the itch impulse ascends along the spinothalamic tract to the thalamus and proceeds to the somatosensory cortex. Itch is thus perceived in the brain and can trigger the desire to scratch. Scratching is mediated by motor neurons and can be voluntary. However, the itch-scratch reflex also occurs in the spinal cord, which can commence independently of any awareness in the brain. This general sequence occurs for any itch sensation, but the specific signals and receptors involved can differ. The other itch pathway I mentioned, non-histaminergic itch, is primarily involved in chronic itch and is elicited by nerves that express a variety of receptors that are activated by pruritogens released by effector cells such as mast cells, basophils, macrophages, lymphocytes, and even keratinocytes. The released pruritogens include alarmins like TSLP and IL-33, type 2 cytokines such as IL-31, peptides, for example, BAM822 and substance P, proteases, extracellular matrix proteins like periostin, and other mediators such as leukotrienes. Once these pruritogens bind to their receptor, the overall sequence of events is similar. However, the diversity of receptors suggests that numerous molecules may reflect differences in terms of itch quality, for example, acuity and intensity. Chronic itch can drive persistent scratching responses associated with skin barrier disruption, leading to activation of immune cells and promotion of further inflammation. In chronic pruritic inflammatory skin conditions such as AD, PN, and CSU, IL-4 and/or IL-13 can directly stimulate neurons and cause neuronal sensitization, or in other terms, increase the sensitivity of itch processing neurons in the skin and promote further itch and associated scratching behavior. However, the array of other factors that cooperate with these cytokines to promote itch differs across these conditions. Further, the spatiotemporal sequence by which various cells and mediators act on proreceptors to trigger itch likely differentiates the clinical presentation of these conditions. I've just talked about immune cells signaling to neurons, but neurons also signal to immune cells, resulting in bidirectional crosstalk. This crosstalk results in not only itch, but also neuroinflammation that shapes the skin's inflammatory response via the release of neuropeptides, such as calcitonin gene-related peptide, or CGRP, and substance P. Keratinocytes, immune cells, fibroblasts, and sensory neurons are in close proximity and likely engage with one another, causing bidirectional crosstalk to become multilateral crosstalk, which may promote the induction and amplification of the chronic itch, scratching, and lesions that we see across chronic pruritic inflammatory skin conditions, such as AD and PN. Similar crosstalk between mast cells, eosinophils, and sensory neurons also have been implicated in CSU. To summarize, in chronic pruritic inflammatory skin conditions, a cycle of itching and scratching can be initiated via an epidermal stress response involving the release of alarmins, proteases, or periostin, or via dermal inflammatory response involving the activation of immune cells and release of cytokines, histamine, and proteases. Immune and skin cells stimulate and sensitize neurons. Neurons release neuropeptides that directly signal to skin and immune cells, further activating type 2 immune cells and increasing production of cytokines and other effector molecules that exacerbate inflammation and perpetuate the itch-scratch cycle. The origin of itch and the cells and signaling molecules involved in it can result in qualitative differences in itch in different disease states. Although AD, PN, and CSU are linked by specific combinations of type 2 inflammatory elements, they are distinct diseases associated with different types of itch. Later in the series, we'll explore what makes itch in these diseases distinct, specifically how the type of signaling involved dictates the characteristics of itch and the ways in which the signaling is dysregulated in each of these three conditions determines the disease-specific nature of itch.